hi everyone. Good day to you all, especially to you, Sir Jether. This is me, Gracious Gia Maglinte, a second-year BS Ed Science student, which we're going to discuss about the Chapter 9, all about gene expression and gene regulation. But in this topic, my part was just all about the overview of the gene expression and the gene regulation. And the gene expression and gene regulation of prokaryotic and eukaryotic cell will be discussed by my co-reporter, Mr. Jerome Lizada and Ms. Rohelda Calles. So let's start. So here's something amazing. Every single cell in our body has the exact same set of DNA. So why is the heart cell so different from the skin cell? Why do some cells grow fast when you're a kid but slow down as you get older? And the answer lies into two key ideas we're going to explore today, which is the gene expression and the gene regulation. But before starting our formal discussion, we have here first our learning objectives. At the end of this section, you will be able to first define gene expression and outline its two key stages, which is the transcription and the translation. And second, identify the four main levels of gene regulation and explain why regulating gene expression is important for our cells. So I hope that at the end of this lesson, we are going to attain all of these learning objectives. In this chapter, we'll break down how genes are converted into proteins and what factors control their activity and how these processes work in both simple organisms and as well as humans. So let's jump right in it. Let's start. So first we have here the gene. So as you can see, gene is a part of DNA that codes for the particular protein, and DNA is the information database of the cell and exists within the cell nucleus. It carries all the important genetic instructions that produce proteins required by our cells. So as you can see here, inside the cell we have the nucleus. And inside the nucleus, we have the chromosomes. Chromosomes composed of DNAs. And inside the DNA, we have a gene. So, in DNA, there are many more genes. And each gene has a set of instructions that is from the DNA. And DNA is the information database of the cell that is located in the nucleus. And it carries genetic instructions to produce essential protein. So as you can see in this slide, the most basic unit of the topic is all about the gene. It explains the DNA that is not just a random molecule, but a structured database stored safely in the nucleus that holds the blueprints for everything your body needs to function. Each gene carries a particular set of instructions, which is usually in a coded format used for an accurate function or a distinct protein. So I have here an example to make you understand about gene and DNA. So you can compare a gene to a specific recipe in a massive cookbook, which is the DNA. DNA is the massive cookbook that composed of many recipes. So DNA composed of many genes. And while the whole cookbook contains thousands of recipes, a single gene is the specific set of instructions needed to make just one thing, like a chocolate cake, or that is what we call the protein. So the seed genes are first transcribed into mRNA or the messenger RNA and then get converted into a polypeptide chain. A polypeptide chain is then converted into a protein. So all the hidden code inside our genes emerge as our physical trait, which are known as gene expression. So what is gene expression? Gene expression is a process of turning on a gene to produce RNA and protein that is called gene expression. Whether in a simple unicellular organism or what we call the prokaryotic cells, or a complex multicellular organism, which is the eukaryotic cells, each cell controls when and how its genes are expressed. So as you can see here in the illustration, it has two main stages, which is the transcription and the translation. So during these processes, I will explain later on. As I've said earlier, a gene is a segment of DNA that acts as a blueprint of making a specific protein. Gene expression is the entire multi-step process from turning on the gene to the final production of the functional protein. So, as I've said earlier, gene composed of the code, the genetic code that is very important in our body. And the process of turning it on is what we call the gene expression. And for this to occur, there must be a mechanism to control when a gene is expressed to make mRNA and protein, or how much the protein is made, and when it is the time to stop making that protein because it is no longer needed. Basically, the gene expression serves as the flow of the genetic information, which is from DNA, and then through transcription, it became mRNA, and then through translation is nakahimo siya og protein. And that is the process of gene expression. So, this activation leads to the creation of messenger RNA, or what we call the mRNA for short, which acts as a template for building the proteins. Simply, gene expression is the flow of genetic information from gene to protein. So, gene expression is a vital process in cells that ensures specific proteins are created at the right time and in the right quantity. So, for example, think of your DNA as a massive cookbook, or which is the genome, that is stored in the library, which is the nucleus. So, each recipe is a gene. Gene expression is the process of picking a specific recipe, like a chocolate cake, writing it down to the separate piece of paper, which is making of mRNA, and taking it to the kitchen, which is the ribosome, to actually bake the cake, which is the protein. So at the end, makahimo sila og product, which is the protein, through that process of gene expression. So now let's move on to the two key stages of the gene expression. So first step is the transcription. So transcription is the first step in gene expression, where the genetic information encoded in DNA that is transcribed into mRNA, or the messenger RNA, by an enzyme called the RNA polymerase. The process of transcription starts with the binding of RNA polymerase to a specific region of DNA molecule called the promoter. The promoter signals the starting point of the transcription. So basically, this transcription is the first step where the information in the specific segment of DNA is rewritten, or kumbaga kinapi lang, dinuplicate lang into a portable molecule called the messenger RNA, or the mRNA. This process is led by the enzyme called the RNA polymerase. So RNA polymerase, which is bind to the start signal, which is known as the promoter. So si RNA polymerase is an enzyme na makakatulong in order to generate or emerge that certain code into proteins. So RNA polymerase unwinds the DNA double helix near the promoter region. So as you can see here in the initiation, the first step of transcription, we can see that RNA polymerase is binding to the promoter. And this allows one of the DNA strands to act as a template for the synthesis of mRNA. As RNA polymerase moves along the DNA template, as you can see here in the second stage, which is the elongation, the RNA molecule is synthesized into 5 to 3 direction, complementary to the DNA template strand. So, transcription continues until a termination, or what we call the third step of the transcription signal, is reached to the DNA sequence. So, the signal causes RNA polymerase to separate from the DNA template, and the mRNA molecule is released. As you can see here in the third step of transcription, which is the termination, makikita natin na si mRNA is ni-release na ni RNA polymerase because nakarating na siya sa stop signal. That means to stop the process. So dito is na-separate na siya kay RNA polymerase, which is ready na siya to occur to the next stage, which is the translation. So now let's move on to the next slide, which is the prokaryotic cell and the eukaryotic cell during the transcription. So as you can see here, in prokaryotes, transcription and translation occur in the same compartment. So kumbaga, yung dalawang process ng transcription and translation is iisang lugar lang yun nangyayari sa prokaryotic cell or the unicellular cell, just like organisms and other mga bacteria. But in the eukaryotes or the eukaryotic cell or the multicellular cell organism, which is us humans, animals, plants, and others, the initial RNA transcript or the pre-mRNA undergoes modification before becoming a mature mRNA. So makikita natin na complex talaga yung process when it comes to eukaryotic cell. And this modification include copying, splicing, and adding a poly A tail to the mRNA molecule. So, in complex cell, which is here in the eukaryotic cell, is that the initial copy is edited before use. So, kumbaga, dili na siya katong original copy sa mRNA. This involves splicing, where non-coding junk parts called the introns are removed. So during this process of transcription in eukaryotic cell is that nire-remove nila yung mga non-coding na part or yung junk part ng mRNA, which called the introns. And the useful coding parts or exons are joined together. So kumbaga, ginakwaan nila or gina-edit nila ang mRNA nga na-produce during transcription. Also, once processed, the mature mRNA molecules of the eukaryotic cells are transported from cell nucleus into the cytoplasm, where they serve as templates for protein synthesis during translation. So there's difference, diba, between prokaryotic and eukaryotic cell in terms of the gene expression. So sa first stage, which is the transcription, is magkakaiba na sila. As I've said earlier, diba, the prokaryotic cell and just happen in the same compartment of the two steps, or the transcription and the translation is nangyayari lang siya doon sa same compartment or sa iisang lugar. But when it comes to eukaryotic cell is may pagkakaiba na sila, because dito is ini-edit muna nila yung messenger RNA before proceeding to the next stage of the gene expression process. So now let's move on to the second step or the second stage of the gene expression, which is the translation. So translation is the process that uses the genetic information encoded in mRNA to synthesize a specific sequence to amino acid, forming a functional protein. Translation begins when the mRNA binds to the ribosome, which recognizes the start codon or the AUG on the mRNA, marking the initiation point for translation. So basically, translation is what we call the building stage. So as you can see, diba kanina is the transcription is just a copying stage. So translation is the building stage. This is the second stage, of course, in the cytoplasm, where the mRNA, which is the message, is read by the cellular machine called the ribosomes. So the ribosome reads the code in 3-letter words, which they called as codons. And then this uses them to assemble into amino acid into a long chain. So if we're going to explain it in a simple manner or in an understandable manner, this is like a chef. Ribosome is like a chef that is reading a recipe, which is the mRNA, and to assemble a specific ingredients, which is the amino acid, that is in correct order to cook a recipe or to cook a meal, which is the protein. So basically, si ribosome yung magiging chef na magbabasa ng recipe, which is the mRNA, that is to assemble a specific ingredients, which is the amino acid that can use to cook a meal, which is the protein. So that is the basic understandable example for you to know about the process of translation. The translation process occurs when the mRNA binds to the ribosomes, which is recognized as the start codon or the AUG on the mRNA, marking it in the initiation point for the translation. So as you can see here, in the initiation process, which is the first process of translation, is makikita natin na si mRNA is nagbind siya doon sa ribosome, and also the transfer of RNA or the tRNA molecules, each carrying a specific amino acid, match their anticodon sequences with the corresponding codons on the mRNA. So during elongation, which is the second step, the ribosome moves along with the mRNA, and each codon is matched with the appropriate tRNA anticodon. Also, amino acid is carried by RNAs to join together a specific sequence, forming a polypeptide chain. So as the ribosome moves along with the mRNA, this chain grows, creating a primary structure of protein. So kapag nabasa na ni ribosome each of the codons, is magkakaroon or magpo-formulate into a chain or long chain that is hahaba ng haba as the ribosome reads all the codons. So translation continues until a stop codon, which is the UAA, UAG, or the UGA, is encountered on the mRNA. So that is during the termination. Stop codons do not code for any amino acid. Instead, they signal the end of the translation process. So kapag narating na nila yung stop signal, is basically titigil na yung process during sa translation. So basically, that is the process for translation. That is, and this part of the process is makakabuo na sila ng proteins that is responsible for all of the informations in our body. So after the translation, the newly synthesized protein may undergo various post-translational modifications, and this modification can include folding into a specific three-dimensional shape or the 3D shape, addition to the functional groups, or cleavage of specific segments to activate the protein. Once the chain is built, it must fold into a specific 3D shape or are chemically modified to become a functional protein that can actually do work in our body. So that is the process of translation. And to sum it up, during transcription, mRNA was released, and during the translation, protein was created. So if transcription or translation fails, the cell might not get to the right point it needs. So this could cause small issues and make cell work wrong, or it die, which is called apoptosis or the cell death. It also leads to genetic disorders like a sickle cell anemia or a cystic fibrosis, or the raise of the risk of the diseases such as cancer. So translation and transcription is very important in the process of gene expression in terms of our genetic informations. Now let's move on to the gene regulation. So what is gene regulation? As you can see, gene regulation is how a cell controls which gene out of the many genes in its genome are turned on or expressed. Thanks to the gene regulation, each cell type in your body has a different set of active genes, despite the fact that almost all of the cells in your body contain the same exact DNA. These different patterns of gene expression cause your various cell types to have a different sets of proteins, making each cell type uniquely specialized to do its job. Not all genes are needed all the time or in every cell. Regulation allows cells to be efficient, not wasting energy or making unnecessary proteins, and allows for cell specialization. Every cell in your body has the same DNA, but regulation makes a heart cell different from a brain cell by only turning on the relevant genes. So, the regulation of gene expression conserves energy and space. It would require a significant amount of energy of an organism to express every gene at all times. So, it's more energy efficient to turn on the genes only when they are required or they are needed. So the control of gene expression is very complex or extremely complex. Why? Because malfunctions in this process are detrimental to the cell and can lead to the development of many diseases, including cancer. So just like for example, all of your cells have a gene for hemoglobin, which is it carries oxygen, diba? Hemoglobin carries oxygen. But only your red blood cells actually turn it on to express it. So your skin cells have the same gene but keep it turned off because they don't need to carry oxygen. So basically, gene regulation is the control of the gene expression. In simple example, to compare gene expression from gene regulation is that when we say gene expression is when your liver cells making a protein that breaks down alcohol. And in gene regulation is that if your liver cells increasing how much of that protein they make only when you drink alcohol, so that is not wasted when you are sober. So in other way or in a simpler manner, expression or the gene expression is the action of making the protein, and the gene regulation is the brain that directs that action. So kumbaga, si expression ang action mismo, and the one who direct o kinsa ang magsugo or maistorya kung kano sa on or kano sa buhaton ng action is the gene regulation. Let's move on to the stages of gene regulation or the levels of gene regulation. So we have here first the transcriptional regulation. So transcriptional regulation involves genetic interactions with specific DNA sites and epigenetic effects. Transcription occurs to interact with DNA, enhancing or inhibiting RNA polymerase binding, which is the epigenetic modifications like DNA, methylation, and histone alterations change gene accessibility without changing the DNA sequence. This is the most common form of regulation and happens at every beginning of the process. It involves transcription factors, which is proteins that bind to DNA to either help or block the enzyme. RNA polymerase from starting of the creation of the mRNA, it is also includes the epigenetic modifications such as the DNA methylation, which can hide a gene so it cannot be read without changing the DNA's actual sequence. So just like for example, in your body, the gene for making hemoglobin is transcriptionally active only in the red blood cells, right? So in skin cell, this gene is hidden by epigenetic marks, so the cell doesn't waste energy making blood proteins that is not needed or that is not required to use. Another example is that if your body doesn't need more insulin, so it stops the insulin gene from being copied into mRNA, so no insulin process even starts. So it means there's no wasting of insulin in your body because it is not yet required or not yet needed. So next we have the post-transcriptional regulation. So gene expression can be regulated by post-transcriptional mRNA processing. In eukaryotes, transcribed mRNA undergoes splicing and other modifications that protect the ends of the RNA strand from degradation. Splicing removes non-coding segments called the introns and joins in the protein coding called the exons. Once the initial mRNA or the pre-mRNA is made, it must be edited before it can be used. This involves splicing where non-coding regions called introns are removed and coding regions like exons are joined together. So this stage is also includes adding a protective cup or tail to ensure the mRNA survives its journey to the ribosome. So after mRNA is made but before it is used for translation, this post-transcriptional regulation happens. This is like editing the film footage before it's turned on into a final movie. So the cell modifies the mRNA, just like for example cutting out the introns, adding protective cups, or decides if the mRNA is even allowed to leave the nucleus to be translated. So basically during this process or this time is gina-check nila if pwede ba mo sulod or allowed ba nga mo leave si mRNA sa nucleus to be translated. So there are many requirements needed during this post-transcriptional regulation. Just like, for example, alternative splicing. So alternative splicing allows a single gene to code for multiple different proteins. So just think of like a modular toy. By choosing different exons to keep, a single gene in your brain can produce a different version of a protein than the same gene in your muscles. So another example is that some mRNAs are locked in the nucleus until the cell gets into the signal to release them, so they're ready but not used yet. So now let's move on to the next stage, which is the translational regulation. So translation can be regulated by the microRNAs that attach to a specific mRNA sequence, blocking translation initiation or causing mRNA degradation. Additionally, translational repressors can attach to RNA and interfere with translation initiation. So various other factors, including RNA binding proteins and specific sequence elements in the mRNA, can also influence the process of translation. So even if mRNA is present in the cytoplasm, the cell can decide whether or not to actually build the protein. So small molecules called the microRNA is attached to the mRNA and act like a physical roadblock, preventing the ribosome from reading the code or causing the mRNA to be destroyed entirely. So the microRNA is isa siya sa tumutulong sa mRNA, and it is act like a roadblock. So it will prevent the ribosome to read the code that is causing to destroy the mRNA entirely. So I have here an example is that during embryonic development, a cell may have many different mRNAs ready to go, but it uses translational repressors to keep them quiet until the exact moment the embryo needs to grow to a specific limb or organ. So basically, translational regulation is a repressor to keep them quiet before that exact action happens. Let's move on to the post-translational regulation. So this regulation is translated by polypeptides that are processed to create functional proteins. Post-translational regulation alters the structure and function of proteins by adding or removing chemical groups like phosphorylation and acetylation, which affect protein activity and gene expression. So basically, after a protein is physically built, it is often needs a finishing touch to work correctly, right? So this includes folding into a 3D shape or adding chemical groups like phosphorylation or adding a phosphate group. That is called as phosphorylation, right? So to turn on or to turn off the protein's activity. So it's either sa regulation if it turn on niya or it turn off ang protein's activity during this stage. And I have here an example from this post-translational regulation. So insulin is initially produced as an inactive long chain, right? So to become functional and manage your body sugar, it must undergo cleavage, where a specific segment is cut off to activate the protein. So it is the job of the regulation whether to activate it or not. Sum it up, the 4 levels or the 4 stages of gene regulation is that first is start the transcription, next step is editing the mRNA, next is make a protein from mRNA, and lastly activate the protein. So that is the basic or the simplest explanation for you to make you understand about the 4 levels of gene regulation. Let's move on to the last, which is the methods of measuring gene expression. We have here first the Northern blotting. It is a hybridization-based method used to determine the size and quantity of specific RNA in a sample. It separates RNA by gel electrophoresis and transfer it into a nitrocellulose membrane where it forms a covalent bond. When we say northern blotting, it is a classic method used to detect a specific RNA sequence, which is RNA is separated by size using a gel and then transferred to a membrane. A labeled probe is used to find and bond with a specific RNA of interest. When we say labeled probes complementary to RNA transcript are used for hybridization. This method is simple and cost-effective, but it is time-consuming and has low throughput. So, in the process of northern blotting, we're going to do first is to take out the RNA. Extract all the RNA from the cells or the tissues, and note that we have to be careful not to break it down. And the next step is to sort it by size. Run the RNA through a gel that separates molecules by how long they are. Smaller pieces move faster than the bigger ones. And next, as you can see there in the illustration, transfer to a membrane. Copy the sorted RNA bands from the gel onto a sturdy membrane so the stray in order are easier to work with. And next is to lock it in place. Use heat or UV light to make sure that the RNA sticks to the membrane permanently. And as you can see there, this next step is to find the target. Add a special probe or molecule that not stick to a specific RNA we're looking for, and the probe should be labeled as so we can see it. And next step is to clean it up. Wash off any probe that didn't stick to the right RNA so we don't get messy signals. And the last step is to see the result and use a machine to film or visualize the probe struck. So the bands show us if the target RNA is present or how big it is and how much there is. So an example of this is that a researcher might use northern blotting to see if a specific viral RNA is present in a patient's tissues sample to confirm if it is an infection. So mostly or commonly researchers use this method to see different infections on the RNA sample. Now we have here the second one, which is the quantitative polymerase chain reaction or the qPCR. So it measures a gene expression in real time by converting mRNA or the messenger RNA to a complementary DNA or the cDNA and amplifying it using the PCR or the polymerase chain reaction. Fluorescent labels are added and the fluorescent is measured after each cycle, which provides quantitative data. So this is a highly sensitive method that measures gene expression in real time. So it converts mRNA into cDNA and then amplifies or copies it into millions of times. Fluorescent labels track how much DNA is made after each cycle, giving very precise numbers, and this method is easy to use and has a relatively short assay time. But, however, it requires prior knowledge of the target sequence and has limited throughput. So as you can see in the slide, that specific machine is the machine used by this kind of process, and this is the technology used in COVID-19 PCR test. It detects even tiny amounts of specific genetic material and quantifies how much of it is in the sample. Today's generation, this kind of machine can detect if you are affected by the COVID-19 or not. So, basically, technology really evolving in this generation. Let's move on to the third methods in measuring gene expression. So we have here the microarray. So microarray measures gene expression by hybridizing cDNA or the complementary DNA strands to quantify transcripts. Microarrays use oligonucleotides or the cDNA attached to a chip surface. So it allows high throughput analysis. Microarrays require separate preparation of control and test samples. So, because this is a high throughput method, meaning it can test thousands of genes at once. Diba? Imagine, thousands of genes at once. thousands of DNA spots are attached to a small cup. As you can see here, it is being compared, the healthy cell and the pathological cell. So, when the sample is applied, the mRNA hybridizes or sticks into the matching spot, allowing the scientists to see the entire expression profile of the hybridized cell. So, as you can see here in the side of our presentation, we can see here the what they call the expression profile, or the result of this kind of method we used. So, it is the DNA microarray. So, makikita natin that this gray one represented that it is not present in both cells. Yellows represented from it is present in both cells. Green means it is in normal cells only, and in red, it means that it is present only in the pathological cells only. Now, doctors can use microarrays on a tumor biopsy to see which genes are overactive, helping them determine exactly what type of cancer it is and which drug will work best. So imagine, microarray is very useful in terms of medicine and the medical field, because it is one of the machine used by the doctors to know if the patient has the cancer. Last but not the least is the RNA sequence directly sequences DNA. This measures expression by counting sequences, which is RNA samples are processed into libraries, sequence, and the data is analyzed to determine gene expression levels. It provides a higher range than microarrays and can detect virus RNA types. So this is one of the most modern and powerful tool. Instead of looking on the specific known genes, it sequences every piece of RNA in a sample and counts them. So instead na tinitingnan lang yung specific genes, is dito sa process na ito is they sequences every piece of the RNA in the sample at binibilang nila ito. And it is more accurate than microarrays and can find new undiscovered types of RNA, but it comes at a higher cost and requires small computational resources. So, makikita natin from definition itself, diba, that it is the most modern and powerful. So, mas eligible siya, mas effective siya in terms of knowing specific DNA or the gene expression. But the but here, however, it is a high cost or need natin ng mas malaking pera or budget, and it requires more computational resources. So basically, this kind of methods or process is applicable lang sa yung may kaya or yung may mga pera. And scientists use this RNA sequence to study rare diseases. By sequencing all the RNA in a patient's cell, they can find unusual mutations or glitches in gene expression that our traditional tests might miss. So that's it, I guess, all about the methods of measuring gene expression. Reporters on a different video presentation. So I guess that's all. Thank you, everyone.